Welcome to Halteres Presents, a brand agnostic interview podcast that seeks to objectively highlight the happenings within the world of diagnostics. And now, your hosts, Rich Thayer and Mickey Yode.
Hello, and welcome to Halteres Presents. My name is Rich Thayer, Managing Partner at Halteres Associates.
And this is Mickey Yode, the founding partner of Halteres Associates.
Today, we are interviewing Dave Persing, Executive Vice President, CMO, and CSO at Cepheid. We've both known him, Mickey, like you, and 30 years.
Yeah. In fact, I've known Dave since our days together as postdocs at UC San Francisco many years ago. Dave's experiences go all the way back to the very beginning of molecular diagnostics in infectious diseases. He's had a great many experiences that we discussed with him in our interview.
And now our interview with Dave Persing. Hello, Dave. Good morning, and welcome to the Halteres Presents podcast. Please tell our listeners a bit about yourself.
Yeah, good morning, Rich and Mickey. It's great to see you again. Yeah, so, I'm a California native, grew up in the Bay Area, going to San Jose State University and then applying for medical school. While I was at San Jose State, I veered off into a side project in which I went to Guatemala to work in a project that involved rebuilding a school after some earthquake damage in Guatemala, and met a physician in the little town that we were in, and I ended up spending a lot of time with him, assisting him in some of his clinic activities, and he actually did surgery in the clinic as well and delivered babies. And so I had a chance to really get exposed to sort of what healthcare was like outside the United States, and was exposed to the impact of infectious disease in a big way in that clinic location as well. So that drove a lifelong interest in the impact of infectious disease, and went to medical school at UC San Francisco, joined the MD Ph.D. program, the medical scientist training program there, and did 2 years of medical school, and then went off to do a Ph.D. in genetics at UCSF, and then graduated from medical school with an MD Ph.D. in 1988, and went to do residency at Yale. And it's interesting that when I was in grad school, I was in the lab doing tissue culture next to one of my colleagues, trainees in the program, and we were both listening to an NPR broadcast on this new method that had been discovered and described called polymerase chain reaction. And I was just sitting there at the bench thinking, that sounds really interesting. I think I might want to specialize in that as a lab medicine pathologist specialist. And that was really the beginning of my interest in molecular diagnostics. So I ended up going to Yale University, School of Medicine, Department of Laboratory Medicine at Yale, where they had a specific interest in supporting emerging technologies, and that really helped expedite my career in this space. I was planning on staying at Yale for a faculty position, but then I got a call from the Mayo Clinic in Minnesota, and wanted to set up the first PCR laboratory at the Mayo Clinic, and ultimately got an offer I couldn't refuse to move to Rochester and build a new facility that was dedicated to doing PCR testing. Those were early days for molecular testing using PCR. Contamination was on everyone's minds back then, because there was no such thing as real-time PCR back then. There was not a closed system for analysis of PCR amplification products. You had to take the amplified targets out of the tube, and you had to then analyze them some way, usually using a blot-based method. And this was just super contamination prone. So we built a special facility to control contamination and keep it out of the areas where we were preparing samples, because that would be the risk of getting amplified DNA from one laboratory back into the clinical samples that you're preparing for analysis. So the special facility was helpful. We also spent a lot of time on optimizing protocols for preventing contamination. So we explored enzymatic methods for destroying the amplicon We looked at isosorelin methods, space methods that would cross-link the DNA, a single strand of the DNA, to prevent reamplification of the target. We implemented all of those in the early days of PCR lab implementation, and we were quite successful in implementing early versions of molecular diagnostics tests we now take for granted, such as tests for HIV, quantitative tests for HIV, tests for hepatitis C, a whole menu of tests that we developed in those days in the Mayo Clinic, and that turned out to be a big benefit for Mayo through their reference lab network, because they managed to advertise the availability of these tests for all of their reference lab clients, and so this went viral as a bad pun, but it went viral and was a big benefit to Mayo and certainly for Mayo Clinic patients as well. A lot of those early activities at the Mayo Clinic planted the seeds for my interests later in life. And we were among the first to be able to show that we could go to a patient specimen of sputum being evaluated for tuberculosis and directly amplify a drug resistance gene directly out of the sputum to predict antibiotic susceptibility or resistance to rifampicin, which is one of the major drugs used in treating tuberculosis. So in 1993 and 1994, we were able to show we could go directly to patient specimens, and using nested PCR, we could amplify rifampin resistance locus and actually predict drug resistance without waiting for cultures to become positive. And that was a huge time saver, right? And that planted a seed for a later interest that ultimately led to development of a test that could go way outside of the reference lab system and have global impact. I would also mention along the way that because of all the publications that we had generated on contamination control, that I got a call, and a man introduced himself as Barry Scheck, who was on the defense team for O.J. Simpson. And this is in 1994. And, he says, hello, Dr. Persing. My name is Barry Scheck. I'm part of the team involved in the O.J. Simpson trial. I'm on the forensic side, and we've seen all your publications on how to control false positives and contamination for coming out of PCR-based methods. And our client has had his samples analyzed by the LAPD forensics laboratory, and we have some concerns about whether contamination could explain some of the results. And so I was kind of, surprised, obviously. I played along. I didn't take sides. But at the same time, I was reluctant to get pulled into a highly public situation. So agreed to review evidence in written form, but didn't agree to take the stand in the trial. And felt like it was kind of be a no-win situation. But bottom line is, I did review piles and piles of evidence from the trial and provided written testimony. And I still have a lot of that correspondence in a file folder in my cabinet in Sunnyvale, California, from that trial, which is of perhaps historical interest, especially now with OJ's recent passing and all the discussion about the trial and whether he was guilty or not. But that was an interesting side story that came my way as a function of doing a lot of publication and providing some level of leadership on the issue of PCR contamination. So I never want to be called the king of contamination, but master and commander of contamination. But that was kind of what I got stuck with in those early days. The biggest impact of any development in the PCR world was the development of real-time PCR, which allowed for closed system monitoring of the PCR reactions inside the tube, so they didn't have to be taken out of the tube to be analyzed, and that enabled a lot more labs to be able to implement PCR technologies without as much of a contamination risk. So that was one of the first steps toward the democratization of PCR technology, making it available to a lot more labs. And we were very much involved in the rollout of that technology as well at the Mayo Clinic. So, that was sort of the first phase of my career, was working in academia. Ultimately decided to move to Seattle to work for Corixa. And that was an exciting time. It was challenging going from academia to industry. In academia, you work on a grant-funded project with a couple of people. With renewal of the grant, you're cycle after cycle for a long period of time, maybe 20 years. You go deep, and for a long time. In industry, you don't have the luxury of time. The model is typically to put, instead of 2 or 3 people, put 20 or 30 people onto a project and go only so deep as to mine value, and then move on to the next project with that team of 20 or 30 people. So for us, with a sort of mild case of attention deficit disorder, it was perfect. But it took time to get used to that, because you got to work on a whole new problem every few years. And it was exciting learning about the new problem and adapting the skills of your team to a whole new set of challenges. But after a couple of years, I really got to like that model and be able to mobilize teams in that way to generate a lot of information quickly.
If memory serves, Dave, that's when we first met, when you were at Crixa and you came down to Chiron Corporation
Yeah,
to chat about blood screening.
that's right. Yeah, exactly. I remember that meeting. I headed up the discovery research division at Corixa and became the chief scientific officer there. And they were acquired in 2005. So I didn't want to move to Belgium, where their vaccine development group was, and was trying to figure out my next career move when the CEO of Cepheid, John Bishop, he and I had known each other from my Mayo Clinic days. I was the scientific advisor for one of his companies. And he called and said, "Hey, I want you to join the board of directors at Cepheid." And so I joined the board of directors. And after my first meeting as a board member at Cepheid, flew back up to Seattle in 2004 and told my wife, I think this company is going to change the world of molecular diagnostics because of what they're doing. They're able to completely automate the process of all the steps that we did at the Mayo Clinic in 4 different laboratories and high containment facilities with an army of specially trained personnel and PhDs. They can do in a little plastic cartridge, and almost anybody can do it. What a breakthrough. So just like real-time PCR helped democratize PCR in the 90s, it seemed like to me that Cepheid's automation, leveraging real-time PCR inside of an automated system, was going to be the next stage of democratization of molecular methods, making it possible not just for laboratories to run the method, but for the technique to be performed almost anywhere by almost anyone. And that would be the ultimate demonstration of the ability to democratize the technology, to bring it closer to patients where the impact will be the greatest. Reference labs are great. I grew up in a reference lab. I built a reference lab, but they're not particularly fast. But really, to maximize the impact, especially for treatable infectious diseases, we needed to have faster results closer to the patient. And that's what Cepheid was going to be able to do. So when Corixa was acquired, I signed up to go to Cepheid, and that was the beginning of my Cepheid career. So I've been to Cepheid ever since then. And it's been the longest place I've been anywhere. And it's been gratifying to be part of really the democratization of molecular diagnostics, which really started with Cepheid, and now we've got lots of companies that have been pursuing the same approach. Cepheid was really the OG, the original gangsta in the decentralized PCR world. It's been gratifying to see where that's gone over time.
Wow. Thank you, Dave, for that introduction. It's a fascinating history of the early days of molecular testing and a reminder of just how difficult such testing was to perform back then. Stepping forward to today, we're hearing a lot about outbreaks these days, be they in humans, farm animals, birds, crops, other plant life, etc. Commonly cited reasons include changes in weather patterns, spillover events, and the so-called shrinking of the planet due to easier travel between far-flung parts of the world. What is your take on these events? How might they impact us all, and where are we headed?
Well, if I had the answer to that, I think it would be probably wrong. But I do think that there's clear evidence of sort of a change in the distribution of infections, perhaps due to climate change. The infections that were constrained to sort of equatorial regions of the world are now expanding into areas beyond equatorial regions, such as malaria in North America now, dengue, chikungunya, Zika. These tropical infections appear to be expanding their range. We've seen the emergence of a new drug-resistant fungus strain, Candida auris, over the past several years that appears to be related to climate change because it's coming from normally much warmer climates is now spreading into areas where it hasn't been seen before from multiple places within these warmer climates. So it's not just emergence from one place, it's coming from several sources. Several different clones, clonal lineages of Candida auris have emerged. So it seems like something's going on globally, not just in one place of the world. The obviously, COVID as a zoonotic infection, this emphasizes the fact that we're interacting with animal species in different ways. The emergence of zoonotic diseases in general. Look at the avian flu outbreak of H5N1 going from mostly confined to birds to now spreading into other mammals. This is a big concern. And who knows what's behind all this, but it seems like there's been a lot more of it recently, and I think it's something to be concerned about. It's easy to become complacent after COVID. Now that the pandemic's over, we don't have to worry. But I think we really need to pay attention to this because it doesn't seem like these pressures are going to go away. And a lot of them are zoonotic pressures, coming from other animal species. So I'm a big fan of the concept of One Health, which views the infectious disease world as a continuum, including animal infections, zoonotic infections, the back and forth travel of infections from humans to animals and vice versa, and the continual adaptation of these infections as they cycle back and forth between species. This is going to be a source of future outbreaks. So, I'm very much concerned about it. I think now is not the time to become complacent. We ought to be putting a lot more effort into pandemic planning and pandemic preparedness. We ought to be designing our tests for the future, and we ought to be designing ways to respond more efficiently and in a more timely fashion than we did with COVID.
You've been involved with a lot of surveillance programs over your career as well. How have they gone? Do we have the tools we need? And should we be doing more surveillance? And if so, how?
Yeah, I think that's a really good question. I think that the interest in surveillance has increased. Never before have we been talking about wastewater surveillance to the degree that we're talking about it now.
Yeah.
Wastewater surveillance really took up a lot of momentum with COVID, and it proved its worthiness, proved its value in the context of COVID in terms of monitoring levels of different viruses that led from COVID to flu A to flu B to RSV to mpox, formerly known as monkeypox. Wastewater surveillance is an incredibly valuable tool for monitoring emerging infectious diseases. And it may not work for everything because not everything ends up in sewage, but it is proving its value for a lot of different applications that many of us would not have considered just a few years ago. But also monitoring the presence of potential emergent pathogens within animal reservoirs. I think that's an important activity to know, to be able to implement and to gain knowledge from, because that will tell us what's out there that could potentially cross over into the human population, and to keep eyes on that and to begin to anticipate the possibility of crossover events by building tools available for doing that, whether it's application of next-gen sequencing approaches or whether it's building tests that are better designed to handle a broad range of targets so that we're not caught shorthanded with technologies that enable us to detect these things as they emerge. This needs to be an integrated approach that allows for better responses the next time we encounter these emerging pathogens. Obviously, working with WHO and Global Virus Network, GVN, other organizations whose job it is to monitor zoonotic infections is going to be really important. And the best way to do this is through international consortia, where we can get international cooperation to allow for these activities to happen in a more open way. Making the data available quickly in sequence databases is incredibly important as well, because that enables vaccine developers and test developers to begin to plan earlier. So we need to have sequence information available as part of this effort as well for planning purposes.
It's so tough to deal with these things logistically, though, sampling the animal population appropriately.
Yeah.
I mean, we don't even know what the sources have been that have caused spillover.
Yeah.
I mean, we have things like eDNA that people are looking at now to
Yeah.
determine what organisms are present and what's within those organisms without even having seen the organism. Are there things you can think of or people are working on to make it more practical to do these kind of surveillance programs?
I think that you're right. It's hard. Knowing even where to start to look for zoonotic sources is often a challenge. I actually had a fascinating trip that I took in 2019 before COVID to Kruger National Park in South Africa, where they were using our tuberculosis test to monitor TB infections in animal wildlife in the park. They were having a problem, and a lot of countries are having a problem with the emergence of bovine tuberculosis in cattle, but also now in other animal species. It's crossed over into other animal species at Kruger National Park. And this has its own burden of morbidity and mortality. But who would have known to even begin testing for Mycobacterium bovis in lions, for instance, or in rhinos or in elephants? And so knowing what to even go after to identify the emergence of a new organism is hard enough. I do think that the emergence of the next-gen sequencing tools that are target agnostic is going to be a big part of the solution going forward. You don't have to guess any longer which reagents to use to go after surveillance. You can actually use these NGS-based tools as fairly broad target agnostic tools to tell you what's there, independent of any specific preconceptions of what might be there. So the combination of broadened surveillance along with NGS, I think, is going to become an incredibly powerful combination going forward. I know many of certainly wastewater testing can be enabled by NGS, but also animal reservoir testing. And that itself may be based on pooled analysis, for instance, of waste samples from cattle, air samples of chicken coops with, for avian flu, that these different sort of aggregated collection activities that don't depend on sampling an individual animal may be useful for applying these methods to get a lot more comprehensive picture of what's actually in the environment.
Yeah, and you mentioned earlier, the crossover of avian flu to mammals, and certainly seeing recently in the news about the occurrence in dairy cows, right, of the highly pathogenic avian influenza. And I think what you're mentioning is the opportunity to look across, food animals, raised animals, farm stock, in much more intensive surveillance programs to see what's happening.
Yeah, and actually, the big surprise I saw this week was the fact that they think now this was a single introduction of an avian flu infection into cattle. From the clonal analysis that's been done, it looks like it was a single introduction that happened much earlier than we thought, like
Hmm.
several months ago, maybe January of this year. So it
Yeah.
takes time for us to even recognize this is happening. But the bottom line is that that particular example is of enormous concern because it's not causing a huge problem in cattle herds now. It's not causing a lot of human infections, but what it is doing is increasing the reservoir capacity of a novel avian strain. And whenever you increase the reservoir capacity, you're increasing the likelihood of additional adaptive mutations that can cross over into the human population. So imagine that, it goes from cows to pigs on a farm, which, has both cows and pigs. Pigs are the mixing vessel for flu because they have both avian and human receptors. And that could be a problem if it gets into pigs and begins to adapt to human receptors. And the likelihood of that happening is increased if it's now in the cattle population, right?
Yeah.
So I think that's what the big concern is, and we gotta watch this closely because we may have to take advantage of those H5N1 vaccine stockpiles in Tamiflu if that happens, and having the diagnostic tools that are ready to recognize H5N1 or H7N9, some of the non-seasonal flu strains, is going to be critical. We can't have tests that are specific for seasonal flu in the middle of a pandemic that's triggered by H5N1 because we will have no diagnostic tools that actually are able to detect it. Even if they're not specific for H5N1, we can't do without the ability to detect it within the existing diagnostic repertoires that we have. So that's a good example of how designing a test is important because it needs to be designed to detect not just seasonal strains of influenza, but also future variants of influenza A, which includes H5N1, H7N9. All of these are influenza A strains. So having a test that can detect influenza A across the board, whether it's seasonal or whether it's avian or whether it's swine flu, we need to have this ability built into our tests so we don't miss it when it comes, if it comes, and when it comes.
Are people doing that with wastewater at this point?
I don't know about H5N1 surveillance. I think there was a mention this morning about detection of H5N1 in Amarillo, Texas. Whether that was human waste being monitored or whether it was intermixed cattle and human waste was a question, but that was a surprise to me to see that. But the bottom line is that our ability to do surveillance needs to be broadened to include H5N1 for sure.
Yeah, a lot of what we're talking about now is really a transition from simply testing humans to testing wastewater, testing animals, testing air, all these other possibilities. And
Mm-hmm.
looking at that, how do you think that's going to develop here? And who's going to pay for it?
Good question. Well, in my example of testing at Kruger National Park for bovine tuberculosis, we actually have two gene experts at the lab in Kruger National Park doing testing for tuberculosis in the park animals. And that's an example of crossing over into veterinary testing with a test originally designed for human testing. They've also been concerned about recent reports that human TB strains are appearing in elephants at Kruger National Park. So it's crossover from humans to elephants. we think, going on at Kruger in addition to transmission of bovine strains between animals at the park. So that's an example of where the technology can be applied outside of clinical diagnostics to other surveillance applications. That who's going to pay for it? Well, it turns out that animals that are positive for bovine tuberculosis can't be exported to zoos. They can't be moved between parks in Africa. So they have to be tested prior to transport to new parks, new zoos, and other places. And that has economic implications because the animals, the parks get paid for animals that are used to be transferred to other parks. So it's a source of revenue for them that they need to protect. And so testing becomes economically important for them to do. In testing in the cattle export and meat export industry is going to be a thing. We think testing for bovine tuberculosis in the cattle and meat export business is going to be increasingly important. Right now, the methods are pretty slow and not very definitive, and so faster methods for testing meat and or animals prior to processing is going to be something that will be justified to be paid for by the export industries themselves because they gain economic value and getting more rapid results. So these are all examples of where this kind of surveillance will come into play in terms of broader surveillance for zoonotic pools of potentially emerging pathogens. I think this needs to be built into, federal budgets and to if it falls under the purview of CDC, I think they need a line item within their budgets to cover it. I think it ought to be part of the global economic picture. The economy of the globe is dependent on having better control over the emergence of new pandemic organisms, and I think there ought to be an interest at the global level in pooling our resources to be able to allow for these activities to happen. And again, I think, between the combination of pooled testing and broad range detection approaches like next-gen sequencing, that these ought to be the approaches that we take because they have the best ability to predict the broadest range of potential emerging targets. This has been a topic of discussion over the past couple of years. Disease X refers to the sort of level of readiness and preparedness we would expect for emergence of a brand new emerging infectious threat from a source somewhere in the world. The example that has been given for disease X has been so far has been Nipah virus. Nipah virus is a measles-like virus that has been documented in outbreaks primarily in India and I think Pakistan as well. And it has a high mortality rate, about 40% mortality rate. It's readily transmitted. We've avoided massive outbreaks, probably more by luck than by direct intervention. But, we look at that and saying, there's just a few steps away from that becoming a global problem. So the wargaming that happens around disease X needs to include specific examples of what we think might be the next threat. And that's one example of how we need to think about planning for an emerging target in the disease X category. They could also be bacterial targets. There could be smallpox-like agents. Mpox was considered and still is considered an emerging target, especially the so-called clade 1 monkeypox, Mpox strains that are in Africa. They have a higher mortality rate. No, they're not transmitted by the aerosol route. They're mostly transmitted by contact and by sexual activity, so they're going to have a different transmission pattern, but they could have significant impact in the short term until they're recognized as a public health threat. So there are many different potential targets that fall into the disease X category, but we need to begin our planning and so-called wargaming tabletop exercises around what we would do how are we prepared to diagnose Nipah virus infections? How are we prepared to diagnose Mpox infections? How are we prepared to build vaccines and antivirals for these things? What's available within our portfolio of antivirals that might be effective? And all these things can need to be part of this exercise to begin to plan for the next pandemic threat, which might be caused by an unknown target, whether it's bacterial, viral, or other. I mean, Candida auris, the fungus, is a big problem. I don't think we're going to see the Last of Us, series due to Candida auris. It's not going to infect the world. It's mostly a nosocomial infection right now in hospitals, but we can't eliminate the possibility that the fungal target might be part of the mix as well.
Absolutely.
We certainly need new antifungal agents, that's for sure.
We sure do. We're running out of them.
Let's just stick with C. auris, which I find to be absolutely fascinating, especially the fact that we have these at least 4 different strains that have appeared,
Exactly.
all of which had antifungal resistant activity immediately without ever having been treated before.
Yeah, I know. They're intrinsically resistant, and that's the scary part. Partly because we've never encountered them in humans before, so we didn't realize we needed different agents to target them. But yeah, it's really a big concern, and we've had outbreaks, of Candida auris now. We've seen outbreaks in Las Vegas, not just on the East Coast anymore. It's spreading. We've seen relatively high colonization rates in patients in long-term acute care facilities who are intubated, where these organisms tend to incubate, and then when they're readmitted back to the hospital, they bring it with them. It's incredibly difficult to eliminate in a hospital environment with standard sterilization methods. Hard to clean up in a hospital environment, so the potential for spread in the hospital is probably greater than for other nosocomial pathogens, healthcare-associated infections. So it's a big threat, and just keeping it out of hospitals and keeping it out of control is going to be a big concern over the next decade. I think it's going to be the turning point for understanding how to control this.
We certainly have the means to do it. I just hope we have the will and resources.
Yeah, exactly. Well, just even doing surveillance for it, patients coming in from high prevalence locations and conditions is going to be the first step, and we're not even there yet. So we need to know where it is before we know how to control
Right.
Good point.
Well, on that happy note.
it. Yeah. It's not the Last of Us series, but it's kind of concerning, right?
Right. Yeah.
Yeah.
It's the last of some of us.
Last of some of us, yeah.
Well, Dave, thank you so much for joining us on this podcast episode. It's been a fascinating discussion. Hats off to you and your career and everything you've done to help further the science and the use for molecular testing.
It's been my pleasure. It's been great spending time with you, and you're right. It's been gratifying, and I think there's more to come.
Wonderful. Thanks so much.
Thank you.
Halteres Presents is produced by Halteres Associates, a US-based bioscience consultancy that provides strategic and tactical services in the areas of diagnostics, medical devices, and life science research to clients of all sizes. The views, information, or opinions expressed during the episode are solely those of the individuals involved, and Halteres Associates is not responsible for any errors or omissions or for the results obtained from the use of this information. The information provided in this episode is for informational or educational purposes only and is not intended or implied to be a substitute for professional medical advice, diagnosis, or treatment. Halteres Associates would like to say thank you to this episode's guest or guests, and thank you for listening to this episode of Halteres Presents.